Now, this week on the Virtual Skeptics, forgot what we were. Bob is not non-GMO. Eve is not stoned. That's good to know. Barb is not tipsy, and Tim is not here. I'm your host, Brian Gregory, and I am all those things. Well, maybe not. One of them I'm not. You need to figure out which one. This is the Virtual Skeptics. Thanks for watching. Hi.
Hello.
Hello.
These are the people who I'll be sharing the next hour or so with. First up,
At least.
It could be a week or so.
It could be a week or so. First up, we have Bob Blaskowitz, Seaside's conspiracy guy, web columnist, blogger for Skeptical Humanities, and Skepticality contributor. Next up, we have Eve Siebert, editor and blogger at Skeptical Humanities, and contributor to Skepticality and Insight at skeptic.com. And we have Barbara Drescher, former psychology instructor, blogger at ICBS Everywhere.
She just perked right up when you mentioned her.
Skeptical projects such as Insight at skeptic.com and The Odds Must Be Crazy. Crazy.
Gravy.
Gravy.
You got it.
Gravy. The odds must be
Gravy. Gravy, baby.
And we also have the invisible, incomparable Tim Farley, who is actually not here, but just gonna.
Where is he?
Well, yeah, we don't know.
Don't say his name. Never mind.
Tim is always with us.
Tim.
It's
It's like something you say at a
every
funeral.
child.
Yeah.
Tim will always be with us in our hearts.
He loved Star Trek.
He is survived by a dog named Blue.
It's true.
Never mind. So, if you're watching and you want to get in on these silliness shenaniganosities, you can via several methods. You can tweet with hashtag Virtual Skeptics. You can comment on the YouTube if you're watching it. Hi, Ben. Or you can actually don't use the Q&A anymore because it's going away anyway. In about 2 weeks, it'll be dead. It will have to be somewhere else and doing other things. And it's just it was nasty anyway. We're not going to talk about it anymore. Okay?
More trouble than it was worth.
Yes. So there you go. So this week we do have a trivia question.
We do.
We do, and it's interesting.
Yeah, so today
Bob is
we
going
saw.
to read the thing.
Yes.
Whether we like it or not.
We saw that there was an earthquake in Italy.
Yes, terrible.
It seems to have killed dozens of people. It was over a magnitude 6, which is severe.
And shallow.
And it hit, yeah, and it hit during tourist season. The cities that were hit were calling with tourists. It's the anniversary of another geological event not too far away. Could you name it? That is our question.
So the question is, today's the anniversary of a geological event in the same area as one that happened earlier today. If you know what that is, please tweet or somehow get our attention. You can send a carrier pigeon to Baltimore, and it probably won't get shot. Dun-dun-dun. Okay.
Okay.
Go,
That was too sad to laugh at. Sorry.
I know it was kind of sad.
Too dark.
Dark. I'm known for my. No, I'm not. I'm not known for my dark humor at all. First, Barbara's first this week.
I am? I
You
thought it was
are.
second.
No,
You were,
it looks
but
like you're
then
listed.
Tim,
Somebody
Yeah.
moved me around?
You've been.
no,
you've
well,
been-
Tim was first, but
Oh, yeah.
Yeah.
Okay, so I've been
you
promoted.
have
Thank you, Tim.
been promoted. So tell us about
Okay.
not being tipsy.
Well, so, I usually get this stuff from my Facebook feed, and once again, thank you, Facebook.
Yay,
and
Facebook.
and thank you, Angie Mackey, who posted this wonderful little link that she said she was not going to even read because she didn't want to be disillusioned. Because it said, the headline said, "Drinking tequila can help you lose weight, science says."
Oh,
I always
Angie?
like it when science says these things.
Yes.
Science
Yes.
says.
The science says tequila can help you lose weight. Now let's see if we can guess whether or not that's a correct or accurate headline.
Technically, if you drink only tequila and eat nothing,
Yeah.
Maybe.
Yeah.
Possibly. Yeah.
Yeah.
Eventually.
And potatoes.
Maybe. Maybe before you die. Yeah. Okay.
Because your liver will fail.
Yeah.
Yep.
Yeah,
Exactly.
Yeah, but you'll be skinny.
And it will
mind.
slowly eat your brain away with a thiamine deficiency, because that's what alcoholism does to you. Okay, so I, of course, looked at the study, and the study is called Agavins Increase Neutropathic or Neurotropic Factors and Decrease Oxidative Stress in the Brains of High-Fat Diet-Induced Obese Mice.
Ah.
Wow.
Tequila.
Do you have slides for this?
I have one. Yeah.
Okay.
The
I did shots with some mice once. Fun guys.
Did you? Yeah, I
No.
bet that was fun.
Not really.
Because you get to drink all the tequila because their shots are like that big, right?
Yeah.
Teeny tiny. All right. So the conclusion on the abstract is agavins may act as bioactive ingredients with antioxidant and protective roles in the brain.
I'm not okay because you have to break this terminology down for me because
Okay.
I don't know what that
Here's
means.
basically the issue. I don't know what I didn't understand a lot of the research, but it doesn't really matter because the parts that I did understand were the important things that tell you this is the headline is bullshit. Okay. So first of all, what is an agavin? And this is a mice study, so take it with that, little.
Take it with a lick of salt,
You're actually asking what an agavin is?
and then some
No,
wine.
I'm going to tell you. An agavin is a fructan, so it is which is a sugar. It's a type of sugar. It's a fermented sugar, FODMAP type. It is actually a type of it's a substance or a sugar that we don't the body doesn't our bodies don't digest properly. They doesn't process. So similar to the way that sugar substitutes work, they're not actually not sugars. They're just sweeteners that our body doesn't process, and so that's why the calories don't, So what they did, so that's what it is. And agavins are extracted from the same plant that you get tequila from.
I was going to ask. It was related.
So
to
there are
agave.
agavins. It is agave. So agavins are from the same plant, so they're in tequila. So that's where you get the tequila connection.
Right.
Now, does it help you lose weight? Well, we don't know if consuming a lot of agavins will help you lose weight. What we do know is that in high-fat diet-induced obese mice, it slowed weight gain. So it did not make them lose weight, okay?
Oh.
What they did is they had 4 groups of mice. One had a standard diet, and 3 groups had a high-fat diet. And then they measured all sorts of things across time or over time. And one of those things was weight gain. And this is where I think the headline comes in. So they had decreased weight gain. The conclusion of the paper is they had decreased weight gain caused by a high-fat diet. So if you will bring up the slide,
Ooh, a graph.
You can see. Yeah, there's a little graph.
Yay.
So the mice that so what you're looking at here is the line across the bottom that has the filled-in diamond that's labeled A is the standard diet mice, which they gained weight over time. So all of the mice gained weight over time. The next one up with the circles are the mice that had that were given a diet of a high-fat diet with agavins, but it was 10% agavins, which sounds to me like a huge amount. I mean, I'm not even exactly sure what that meant, other than I think it was 10% of their diet was agavins. So
wow.
The line above it that has an open triangle.
Mm-hmm.
Those mice were on a high-fat diet, that also was included agavins, but they were in the 5%. And they are statistically indistinguishable from the mice that had the high-fat diet that had no agavins in it. So apparently you have to have a large amount of agavins for this
What was the
effect
point
to
of this
occur.
study?
Uh,
Other than to, like,
Press
stuff
coverage?
mice?
It was no, it was they had a few goals to meet, and I don't understand it well enough to all of their measures, but they looked at
Because it
things like
sounds like
glucose
something that they
levels and all
came up
sorts of things.
while they were drunk. Okay, this is what we're going to
Maybe.
do.
No,
We're going
it
to
does.
force feed these mice and give them tequila.
The
There
thing is, it's not. They're not feeding him alcohol though. They're just feeding him.
No, just agavins.
Yeah.
There is a background. There is a literature.
No,
review
that'll look weird. Just agavins.
There is a literature review that I believe justifies the study, and just the results I think justify the study, because what they're looking at is this particular substance having a protective effect in the sense that it's possible that it's just because it the calories it does something that prevents the mice from eating more because they had decreased food intake, the ones that had the 10% level. And there were other measures in the study that don't address the weight loss thing, so I don't want to go into that. And so there were other reasons to do the study, and there are lots of things that they measured. But the point here is you're not losing weight. They didn't lose weight. They simply had a slower weight gain. They were protected somewhat from the weight gain that would be caused by the high-fat diet. And two, it's not tequila.
Yeah.
It's agavins, which come from the plant that they make tequila from. So agavins will be present in tequila, but drinking tequila has other things attached to it. There's nothing in the study that says if you drink tequila you will lose weight. Absolutely nothing. No reason to believe that. There are too many things that get loaded in with the tequila part that you can't, address. So
just a question. Okay, so
yeah,
if I were to replace 10% of my diet with Fuji apples,
yeah.
Would I not gain
I don't
weight as
know.
fast if everything else was fat? I don't know. Doesn't make any sense to me.
Well,
if you're an obese mouse.
If I'm we'll see.
If you're an obese mouse, but it does make sense that there is something about the agavins that is causing this, because it's not a 10%. I mean, if you look at the 5%, there's really very little difference. There's no statistical difference from them in the high-fat diet. So it's not just that they didn't eat as much. They actually decreased their food consumption. So it wasn't that they were eating the same amount and 10% of it was agavins. They ate less, and that's why they didn't gain as much weight.
Yeah, I was wondering. My real question was, how do they control against other factors that would also affect expected weight gain? I guess is because change the amount of. I don't know. It seemed to me like it was without knowing the details of the study. Yeah.
You make that the only thing. I mean, the only difference between these animals was the proportion of their diet that was.
Yeah, but percentage.
So these are different diets.
I know, but if they're measuring the amount of agavins they're giving them in a percentage, what's that percentage of, and how does that affect their actual food intake? And it just seems like a really poor way of.
But no. When you're measuring everything, you can account for those things, and they did account for those things. So the reason that they didn't gain as much weight is probably because they didn't eat as much.
That's what I'm saying. Yeah. Okay. Yeah.
So statistically, when it was only 5%, the mice ate just as much as the ones that got the no agavins at all. They had to make it 10% of the diet in order for the mice to actually eat less.
10% of what?
10%. I'm assuming that it is 10% of the diet itself. So 10%
Calories,
of the
weight?
Calories, weight,
probably the
weight of
calories.
food.
I don't know.
Yeah,
It was
I
a
mean,
little a
yeah.
little difficult to trudge through, and
Okay.
it was one of those
That was
studies
my
where
question.
it's also one of those studies where there's a lot of biomed's not my field. Where there's a lot of assuming that you're talking of audience that you're talking to, and so they leave, in order to
Yeah.
save space, they leave out a lot.
No,
It was
that
really
works.
kind of weird because the study was had the intro and then the results and then the method after it, which was very bizarre to me because we never do it that way. But part of that I think is to save space, so they get to the point that you're reading, and then
Because all the marketing people aren't going to read
I
past
don't know.
the results.
The methods were pretty short. It's a pretty short study, but I'm not biomed, so
Yeah.
from what I could extract from it, I believe it was 10% of the calories probably, however they do it, but it was 5% and 10%, and the 5% didn't do it. The 10%, it had to be 10%, which means that there's a lot of things out there that could. Things like when they fed saccharin to rats and it caused cancer.
Yeah.
Well, they fed it in extremely large doses for the size of the animal.
Yeah.
So there are things like that where it could happen.
So
This
when we
could have this
soak the
effect, but it
rats
could
in
be-
a solution of saccharin for 5 days,
Exactly. It causes cancer. And this could be one of those things. Yes, agaves could have, some protective effect, but maybe it takes a lot.
and
We
no
don't
doubt.
know.
Yeah.
But my point is that this is a mouse study.
Yeah.
It's and it's a well done study. Don't get me wrong, it's not a bad study, but the interpretation of it should not be, hey, I'll drink tequila and lose weight.
So you're saying the headline is completely accurate in except for everything.
Yep.
Jinx.
Yeah.
Pretty much.
So I mean,
so I don't need to be biomed to I don't need to be in biomed to know that this doesn't line up.
So do you think this was somebody deliberately finding a research topic to get a headline?
That people will share on Facebook maybe and like and promote and
Is it a PhD project? I was curious. Do you know if it was a PhD
I have
project?
no idea.
Yeah, because that seems like it's pretty common. I know with engineering stuff, a lot of the PhD projects end to be something that may not be practical, but is
going to get somebody's attention because you want to be able
gets
to.
Attention. Yeah, maybe possible patent. Yeah. So, okay.
I don't know.
Yeah.
But I found it interesting.
Yeah. Yep.
There is some interesting stuff. I would invite anybody to read the study because there is some interesting stuff about oxidative stress in there too that I don't understand well enough to talk about, but I thought it was interesting.
On a related note, Eve is not stoned.
No?
No, I'm not really sure what you're talking about. Is it actually stones, or is someone being killed by stones?
No. Well, you'll just have to find out now, won't you?
I'm on edge.
This is actually sort of a follow-up to a story I did about almost exactly a year ago, less like a week or so. I talked about exciting new discoveries concerning Durrington Walls, a super henge that's a little under 2 miles from Stonehenge, and probably related to Stonehenge in some way. And I'd like to take just a brief moment to talk about the word henge because I find it fascinating. Henge is actually a back formation from Stonehenge. Stonehenge has been called Stonehenge for a very long time, or Stonhinges or Stonehenge, at least from the early 12th century. No one's entirely sure what henge originally meant. It may be related to hang. There's a theory that the trilithons or trilithons, the massive stones that have still lintels across the top, reminded people of huge gallows. There's also an Old English word, Hengecliff, which is used to gloss a Latin word that means precipice. So it may mean hanging, suspended, or supported stones. It's also been suggested that henge comes from hinge. Again, that would be a reference to the lintels. So we don't really
What's this
know.
have to do with soup?
Good point. I actually saw that mistake in a book where there's a lentil across a doorway. It was hilarious. But
Yes.
anyway,
Soup accidents.
yeah, we don't know exactly what it meant originally, but in relation to Stonehenge, which again is where it comes from, henge had something to do with stones. However, in archaeological terms, it's come to have a relatively precise meaning that has nothing to do with stones. Strictly speaking, a henge is an earthwork with an external circular or oval bank and an internal ditch. Now, Stonehenge's ditch is outside the bank, so it's not a true henge.
The damnedest thing.
So to recap, henge comes from Stonehenge and originally referred to Stonehenge's stones. Now henge refers to earthworks and has been defined in a way that makes Stonehenge, at best,
Not a hinge.
yeah, an atypical henge. It's like
Sorry,
people saying, "Oh,
non-hinge.
that's not really a bug. That's a beetle, not a true bug." It's like, screw you. Bugs have been around before entomologists. You don't get to change that. Anyway.
And that's funny. I was taught that beetles are the only true bugs.
I might have it
Well,
backwards.
it depends on whether or not you want to do a punch buggy or not.
A punch buggy? What the hell's a punch buggy?
Never mind.
Is that like a slug bug?
Maybe.
Okay.
I've never heard of
Is
a
it a
slug
joke
bug.
that involves punching someone?
Yes.
Okay, then it.
Whenever you see a Volkswagen,
buggy?
a punch buggy, no punch back.
What weird country
You
are
don't
you
you
from?
don't know punch
I don't-
buggy, no punch backs?
No.
Please, internet, help me.
No, you're gonna
No.
die on this hill alone.
Okay, fine.
Anyway, Durrington Walls isn't a true henge either. It's a hinge enclosure or a super henge, which means it's
Super
really
hinge.
big and it has superpowers. No, it's really big and there's evidence of occupation within the enclosure. There are a few super henges in Britain, and Durrington Walls is the largest. It's, I believe, the largest Neolithic monument or site, at least in Britain. Now, the big news last year came from the Stonehenge Hidden Landscape Project, led by the University of Birmingham, not in Alabama, in association with the Ludwig Boltzmann Institute of Archaeological Prospection and Virtual Archaeology, also not in Alabama. Not that you'd think it would be necessarily. The project has been using high-tech methods to explore the area around Stonehenge via non-invasively. That whole area is littered with ancient sites. If you can bring up my first slide. Yeah, that's one of the images, possibly stitched together, of the area. Durrington Walls is toward the right, and you can see it's a much bigger circular thing than Stonehenge, which is more toward the middle, or Woodhenge, which is right by Durrington Walls. The next slide is a picture of the bank around Durrington Walls. The white dots are sheep to give scale. Actually, I think they were already just there. They didn't bring in the
one
sheep.
standard
There.
sheep.
Yeah. Lay some sheep down here. We need some scale.
Yeah.
Eh.
One
And-
standard
Disappointing.
Well, the area around has been, cultivated for a long time. So it's
the
really
sheep-
hard to see in some places. Not the sheep. So it's hard to see the bank in some places. There's a road that goes through it, and certainly the bank is less high than it used to be. The earthworks were constructed around 2600 BCE, which is supposed to be a little bit after the Sarsen Circle at Stonehenge was erected. Last year, the project announced that they had discovered what appeared to be a row of around 900 to 100 standing stones beneath the bank at Durrington Walls. These, they thought, had been knocked over intentionally when the bank was constructed. In the next slide, the green dots, sort of at the bottom there, represent the putative stones. And the next slide is one of many artists' impressions of how the stone row might have looked. And these have become so ubiquitous since last year, in fact, that I had a difficult time finding good photos of Durrington Walls that didn't have the stones superimposed on the monument.
Does anyone else wonder if any of these stones move and lust for blood?
Well, yeah, obviously.
Okay.
I'm just wondering what the purpose was if it's not really a wall.
Oh, they're not. They're silicon-based creatures that cleanse.
Okay.
Okay, never mind.
So you
like
know what
Doctor Who reference.
this is for? What these hinges are for?
Mm-hmm.
What?
Ritual.
Well,
I think it's the world's first modern art gallery.
mm,
Not really
Don't make fun
modern
of
anymore, I
ritual,
guess. But
It's the great times 20 grandfather of Christo.
it was not. They didn't cover it. Well, whatever. You never know. Anyway, last year when this news hit the media, none of the stones had been excavated. Since then, two of the features have been excavated. That's the last slide.
hey, look, interns.
Look, they're digging. And guess what?
Look, PhD students.
Yeah. There were no stones.
Hey.
And there are no stones.
Well, why did they think they were there then? Didn't they have?
Because
Sonar-
of the scans from
Yeah.
all sorts of things. It was giving signals that looked like big stones. I'll sort of get to what it actually is
Okay.
there. And now, it may seem like a little bit of a letdown that there weren't stones there. And some of the stories about the new discovery have had just a bit of a tone about them. For instance, a USA Today article begins, "Remember the huge stone monument archaeologists found buried 2 miles from Stonehenge in the UK last year? Well, it turns out that the so-called Superhenge that radar suggested was made of 100 or so large stones was actually stoneless." As if it's slightly embarrassing that
You can
We're not
actually
stones.
detect something that was organic from that long ago. That's pretty amazing.
well, yeah, but really, my feeling
Besides
is
stones. Yeah,
a
go ahead.
hypothesis has been falsified, and someone did a science, which isn't bad. And I think this example also shows the immense value of both the high-tech, cutting-edge, non-invasive archaeological tools and traditional digging. The non-invasive scans would keep improving, and they help pinpoint areas of interest, reveal features that are not visible to the naked eye, even from aerial photographs, suggest what might be there, and help target digs. However, there are still some things that can only be confirmed or falsified by digging, even if it's just a small targeted dig. Now, at Durrington Walls, the pits had been filled with chalk rubble, and then the bank was built over them. And I believe it's the chalk rubble that was giving signals that was
Non-dirt
interpreted
signal.
as,
As a non-dirt signal.
Yeah, as a signal that was interpreted as stones.
Just out of curiosity, in this picture, was this picture used for publication or something, or?
I don't know. It came from.
What is, this is the setup for a joke, right?
Yeah.
It is.
Yes, okay.
There's this one guy in the back with a like his T-shirt must be like Jack Daniels or something.
Yeah.
He's just standing there with his hands in his pants, and I'm trying to figure out what his job is on this
He's the
particular.
urinator, obviously.
Yeah. You know the other guy
Don't
the
ask
other guy-
how.
in the background with the sort of off-white shirt
Yes.
standing.
Okay, so he's actually the one that's peeing.
Actually,
The other guy
I'm the
is kind of
wheelbarrow.
sitting there. What?
Or on her head. I can't tell.
Yeah.
Anyway.
I think I may be wrong about this, but I think that might be Mike Parker Pearson, who may be the lead archaeologist. Bob would know. In a Time Team, he was on several episodes of Time Team, which is an archaeology-based show in the UK, and including one on Durrington Walls, which he had been working at for some time. And he must be thrilled, by the way, with what they actually did find. So anyway, before being filled in, the enormous pits, and they are huge, they had contained massive timber posts. According to National Trust archaeologist Nicola Snashel, the pits, quote, have got ramps at the sides to lower the posts into. However, at some point the posts were vertically lifted out of the pits. I'm not sure how. According to Snashel, the timber monument was erected right around the time that a settlement on the site, which was used by Stonehenge builders, went out of use. Now, The Independent has a long, exclusive story on the new discovery, but it's full of assertions and contains no quotes from experts, so I'm a bit hesitant to trust it. So just keep that in mind. But the article says there was a giant timber circle, and a circle, so it sounds like it went farther than what they thought that row of stones was. On the other hand, they also say it was never completed. It refers to the removal of the posts as a revolutionary religious and political change. A spade from a cow's shoulder blade was ritually placed at the bottom of one of the pits before it was filled in to ritually decommission it. And they linked the changes at Durrington Walls to changes in direction that were also going on at Stonehenge, for instance, moving from smaller stones in a larger circle to those giant stones that are sort of the most familiar aspect of Stonehenge. And at Avebury, which is another monument that's still in the same area, where the circle was expanded. They also link it to the arrival of the Beaker culture, or Beaker people. Yeah.
The Beaker people!
Yeah, I got it. Now, again, just imagine a whole group of people coming to Britain and just wandering around these ancient monuments going.
We lost our shit.
Yes,
Yeah.
totally.
Now, again, I'm cautious about placing too much reliance.
There's just something about Beaker.
Yeah. I'm cautious about placing too much reliance.
Someone needs to do a Beaker. Someone needs to do a Beaker people investigation and byline Dr. Bunsen Honeydew.
If Honeydew and Beaker did archaeology, wouldn't the stones fall over a lot?
That's true.
Yeah.
That's true.
And possibly explode?
Probably would not have a lot of things left, that's for sure.
Yeah.
Beaker would be at the bottom.
Yeah.
Squashed
Along
by
with
a henge.
the shoulder
or a
blade
non-henge,
of a...
actually.
Yeah. No, it isn't. Well, yes. Henge-like. So I'm cautious about placing too much reliance on some of the Independent's bold assertions, although they aren't inherently implausible. I just like to have a better idea where their information comes from. What we do know is that instead of a massive stone monument, we have a massive timber monument. So closer to Woodhenge than Stonehenge structurally. And Woodhenge, again, as you may recall, is actually very close to Durrington Walls. And just decades ago already, 2 timber circles were discovered within Durrington Walls. And since there are two of them and they're within Durrington walls, it sounds like they're, just little things. In fact, there are actually pretty substantial circles um, on a level with Stonehenge, but made of wood. Again, huge post holes and massive hunks of tree. So
Phrasing.
again, a huge amount of effort.
Phrasing.
Yes. A huge amount of effort to do.
Sorry.
Um,
See, meanwhile, in my head, I'm thinking that Woodhenge would be a great Spinal Tap album.
Oh God, yeah.
yeah. Although, again, also it quite large. We don't know exactly why the wood monument was built. There are theories about it, and that is actually related to Stonehenge. One theory is that because there was feasting at Durrington Walls. They buried remains of feasting. They particularly liked young pigs. But the theory is that it was part of the process of death. That there was a celebration at Durrington Walls, and then the remains would have been taken to Stonehenge, and which would have been sort of the end part of them becoming spirits or elders. At any rate,
where were the people involved in these rituals, the dying? Where were they buried?
They may have been buried around Stonehenge. People have been buried at Stonehenge for a very long time. I think that it was a cremation burial site before anything was actually even built there.
Okay, so okay, I didn't know that.
Yeah, and one of the stone circles at Durrington Walls is a line toward the winter solstice sunrise, I think. I don't know. Anyway, and then, okay, so we don't know exactly why they were built. We don't know why the posts were removed and why the pits were covered with massive earthwork. And obviously, since we don't have all the answers, it's imperative that we fill in those empty spots in our knowledge with ancient aliens or some sort of nonsense, because that is how science
not
works.
knowing. Yeah.
Yeah.
We can't have
because let's face it, you can't prove it wasn't.
Yeah.
Yeah, that's a good point too.
I was gonna say aliens are fun, but okay. These are the ones I party with.
Yeah.
When Trump comes to power, they won't be allowed in.
As far as you know.
Now, those would be the only sorts of illegal aliens that are allowed in would be the reptilians.
Oh no! He's going to soften the stance on the people who are already here, which means he's probably going to end up softening the whole immigration thing too. So.
But what about the reptilians?
He's an awful person who will say literally anything.
Huh?
We might be going off track here.
I think we are. God, that
you brought up Trump.
Yes, don't bring up Trump, please.
I didn't bring up Trump.
That's true.
What does that have to do with Stonehenge anyway? Except the fact that you want the stones to fall on him. But
you will be visited by
the almost henge. I don't know.
It's not what it says it is. I'm reaching here.
No, there was the alien.
Eve got what I was saying, but I was reaching. That was reaching. Sorry.
Anyway, Eve, that was awesome. Anything about henges in general? I really find that.
You're a sucker for henge.
I'm a sucker for henge. Give me some henge.
You're gonna put a little henge in your backyard, aren't you?
I am. I'm gonna henge the shit out of
one
it.
that's 6 inches instead of 6 feet.
Yeah, the little tiny one from Spinal Tap.
I'm gonna make it out. I'm gonna have it come
That's my
down.
favorite scene. I love that scene. It cracks me up every time. They slowly fly it down. And
Okay. Moving right along. Yes, I'm doing this. It's been a long time since I played the music, so I wanted to actually enjoy the pounding of robots coming to kill us all. Although this is a good thing. I've only got one story this week, and I'm going to play this neat little video. This is going to be fun. This is a pointless but fun video. Da 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 da. Oh, it's making noise. Can you hear that?
Nope.
Okay, good.
It's like the $6 million robot.
Yeah, last year at the end of the DARPA Robotics Challenge finals, NASA announced a challenge for humanoid robots called the Space Robotics Challenge. This is supposed to be, and then this is in their quotes, to prepare robots for the journey to Mars. Blah, 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 you know, this is pre the Martian, kind of this whole thing that NASA's been talking about for, actually it's been, how long has it been? Isn't George Bush who originally signed the whole exploration? Yeah, no. Yeah, W, who wanted, yeah. So NASA's been kind of refocused on trying to figure out how to explore Mars as one of the things that we want to do out there. And robotics is one way of doing it. So in the vein of the DARPA challenge, since they've been so successful with pushing technology, they decided to do their own challenge called the Space Robotics Challenge. And this is mainly for humanoid robots. And the idea is the first challenge is going to be virtual. So there's actually a robotics software suite that you can get and download and play with it on your own if you want, called Gazebo. It's a simulator to simulate 3D kind of environments, physics and stuff of doing controlled robots. And the final challenge is going to be on the R5 Valkyrie, which is, I talked about in the show before, it was built by JPL. It was supposed to be in the DARPA challenge, but it wasn't quite ready, and I think it wasn't really a priority for them at the time to get it into that challenge because they knew this was coming. This is like their first prototype platform for a humanoid-shaped robot that's supposed to operate in space, either teleoperated or autonomous, which is onboard electronics. What they announced this past week is the initial tasks that the challenge is going to be about, and a little bit about how to enter, because it's supposed to be open to anyone. Although there are some restrictions if your team wants to win one of the prizes, which is if you win first prize, it's $125,000, $100,000 going to second place, $50,000 to third, and $25,000 to fourth. And there are also 6 teams that can take home a $50,000 bonus if they complete all 18 subtasks in one run. Now, these are some of the things this is the scenario that they've built to try to, because like the DARPA challenge, they had kind of this scenario of rescuing in some kind of hazardous environment. Their task is, okay, you're on the moon, the Mars, somewhere. You're in a space habitat, and something has happened. Let's see here. This is the. I'm looking for my notes. Okay. So something has happened, and it has disrupted it has disrupted one of the solar panels, so you're losing power. The habitat itself has an air leak of some kind, and your communication antenna has now been askewed, so you can't talk to Earth. And the robot has to repair the air leak, install a new solar panel, and then fix the antenna. Doesn't this sound like The Martian?
It really does.
Yes.
And I was just like, okay, so when does it start eating its own potato pieces?
And apparently the fourth task is the robot has to cuss profusely and shine shit out of everything.
That's good.
What
That's good.
how long? I mean, what's the time frame for this? Because there's a first, second, third, so there has to be a time frame. It's not a race.
I don't have details on those kind of parameters, although I'm sure there are, and I'm sure probably some of that might be more formalized by the time the actual challenge comes around. Yeah. There are other restrictions related to communication to your robot, and other things that are similar to the kind of restrictions you'd have if you were maybe on Earth or on a spacecraft or somewhere, and you're controlling a robot remotely through space. So the robot is either on the moon or somewhere else. So there's communication delay, and there is a time limitation, 2 hours. But it's not really. I mean, it's not really. I don't think that's a hard problem to solve. I think the harder problem to solve is teaching the robot how to do the basic tasks, which the DARPA robots have done similar things on Earth, and then dealing with some of the environmental tasks or the specific small motor tasks, like repairing, like finding an air leak and repairing it, depending on the size and location and where you have to be to do that. That's a bit tricky.
When I was asking about time frame, I meant the time frame for the competition.
Oh,
how long do people have to develop this? 'Cause
the virtual competition will be June 13th to 16th next year.
Wow.
And we'll know who won by June 30th. That's the first stage. The second stage hasn't been announced yet. Sign-ups are going on now, so anybody who wants to put a team together can actively sign up, and I think it's only about a month you get to register. If you want to win one of the prizes, either your team lead has to be a U.S. citizen, or you have to have a certain amount of your team to be U.S. citizens or enrolled in a U.S. school. So that's because it's NASA. You have to kind of be pro-U.S. That's all the details I have right now. But it's cool. I'm going to keep tabs on it. What?
The question I have is, like, you would so it looks like the idea is to make a robot that's autonomous enough that it can make repairs and keep itself going, even when its communications have been cut with Earth.
Yeah,
right. But there's so many ways that could go wrong. Like the solar panel could, have a short, or it could fall apart, or there's so many different ways to do that.
Or what if the Mars rover gets jealous and attacks it?
That's true.
Actually, I would be more worried about Sojourner. It's a bit plucky.
It's true.
Well, I've read sarcastic rover's tweet.
Yes.
Yeah,
it's some of the specific details are kind of deliberately left open because they want problem solvers. But in reality, it's very similar to the DARPA challenge. There's a lot of the same things. They don't have to drive a car, though. They do have to do more fine motor stuff, and they do have to deal with space environment. So everything that's built now, I don't know if they're requiring this at the hardware level, but there is a implied requirement that it has to be non-terrestrial, which, since it's not in space, it's not as big a problem. See, if you're away from a gravity well, you have no atmosphere at all, you have no protection from radiation, you have to use special hardware, computers, and stuff like that. But if you're on a planet, even Mars and even the Moon, there is a little bit of protection, and it's not as big a deal. But I don't know how they're going to work that into this either. But generally, there's an implied limitation on performance of your computer chips and communications and all kinds of limitations on electronics. But yeah, I think we could probably duct tape and some Arduinos and a couple of LEDs. And
how much fun is that? So these simulations, this is when we went to visit your work.
Yeah.
It looked like they were doing simulated work in a simulated space. This is the same type of idea.
Yeah, it's not the same piece of software. We use ROS, which is a robot operating system. It's a big collection of software that includes some visualization and virtualization stuff where you can model real robots in the computer, so the physics all work, the motors all behave the same way, the sensors all work as if they were within reason, within reason what they work in real life. Gazebo is an open. I think it's open source, and it's not ROS. It's kind of what NASA has been using on the other side. See, we do servicing. There's a whole other branch of robotics on the other coast that does rovers in space, and we talk, but we don't do a lot of the same technology. It has different problems to solve.
So okay, cool.
But yeah, it's the same kind of thing. And the advantage is that a team or anybody can really download Gazebo and start playing with it and learn how to use it and build something virtual in it and maybe win a prize if you're very clever. Do
That
it,
is
man.
pretty awesome.
And all of this, is just bringing us all closer to when the robots usurp us.
And
So-
colonize off planet so that they
You
are.
do realize that the
actually-
colonization
Take
of
all our
robots.
jobs.
Yeah. No, they're going to build a different wall. A wall around the planet.
Ah,
Anyway,
Douglas Adams style.
one can only hope. We are dangerous, but mostly harmless. Great. Now Google Docs says I can't do that. But who's next? Bob? Have you
No.
gone yet? No.
I'll go.
Are you getting stoned or something again?
I'm the last.
No.
That was Eve is not stoned. I'm not against GMOs.
Oh.
So my story today isn't exactly a skeptic story. You could do a skeptic spin on it, if you wanted to. But really, I wanted to clarify something in my own mind, which I think is going to be very important. A lot of people are talking about it. We may have mentioned it on the show before.
I'm terrified.
A lot of people are talking about it. Must be
Bob's
true.
mind.
Must
Yeah.
Who's
be
talking about
accurate,
your-
good stuff
Yeah, who's talking about your brain? I don't.
or smart people talking about it?
Yeah. Really top people.
I'm holding my breath until you all apologize. All right.
People.
So
People.
people are talking about is CRISPR.
Not you, of course.
Not
Just
me.
people.
CRISPR. Those
CRISPR.
little apple chips you get on the side at the deli.
No, it's
No.
where you put your vegetables.
Oh,
Yeah.
yes, the CRISPR.
Right.
Mhm.
Yeah. So they're known as CRISPR is clustered regularly interspaced short palindromic.
Crispin Glover.
What?
Crispin Glover.
No.
Go ahead. I had to
All
throw
right.
one more in.
So, CRISPR is a technology. It's a new technology, a gene editing technology, which is really
Oh, yeah.
precise. It was discovered. The first description of what we would eventually call CRISPR was found in E. coli in the very early 1980s. Japanese researcher, and what he described was this. He saw 5 repeating sequences of DNA. That were separated by short but different spacers in between them. So you would see this pattern perfectly repeated like 5 times, and then in between there was a little different bit of DNA that didn't match anything else in the strand. And eventually they started finding this type of stuff all over in lots of different types of bacteria. They found this repeating pattern. They came up with the acronym CRISPR. And they didn't exactly know what they were. I think that the first, I'm not seeing it in my notes, but the first really big hint about what was happening there, because if it's seen in so many different bacteria, clearly it must be doing something. And what they did, so it was 2005, they had a database where they could cross-reference these genomic sequences against thousands of other species. And what they found is that it wasn't the important bit, it seems, were these little spacers in between the pattern, the repeating pattern. Those most closely matched viral DNA. And what that, it turns out that it's part of a defense mechanism that bacteria have to fight reinfection. And so what happens is that, usually when a cell is invaded by a virus, a bacterium is invaded by a virus, there's a reaction and a defense reaction, a disorganized one, that doesn't really necessarily take hold, and usually the cell will die, but you will occasionally have survivors. And when you do survive this viral attack, the scissors come out, they snip up the invasive DNA, and a little snippet of it ends up built in and stored in the bacterium's own DNA strand, buffered by these spacers, right? And so when they found that these little buffers, these little things were viral, they were realizing. They started to realize what this was doing. When a cell was reinfected, or I guess when its descendants are reinfected by that same virus, there is now a pattern that when the viral DNA is injected, there's a pattern that can be recognized now, and so they send out these little proteins that have little bits of RNA that have to match exactly, and it'll latch on, and then it'll snip that out and remove it, which is very clever, right?
Mm-hmm.
Does that make sense?
Kind of.
What did I fuck up?
I don't understand how this works at all, so I'm trying to follow.
Yeah.
So they insert some kind of pattern into the viral DNA that has to match with.
Okay, so the virus injects its DNA into the cell, and it starts using the cell's resources to make copies of itself, and that kills it. But sometimes it doesn't succeed and is killed by the cell, and then that DNA is cut up, and a little snippet of that DNA becomes incorporated into the cell's own DNA. Okay, and it's buffered by those repeating pads, those repeating patterns.
So it's getting inserted into the cell that doesn't die, the cell that doesn't get killed.
Yeah, it's using its own DNA as storage space, as memory for
The virus.
for viral DNA, right?
Interesting.
And so this my understanding is that this defensive protein that has this little bit of RNA attached to it is what the protein uses to check DNA that it finds floating in the environment, and it will only latch on to something that fits perfectly, right?
Okay.
So and that dispatches the killing enzymes or whatever it uses to destroy the DNA. So this is what they found it does, right? It is a way for the cell to recognize exact patterns of DNA to target, right, and to remove.
Okay.
Let me see.
So how is what does the modification part work?
So how does CRISPR become
modify
something
by
that
putting it in there?
modify GMOs, right? Well, it turns out that once you snip out the offensive bit, the viral DNA that's found its way in, well, what you can do, you can take those enzymes or the proteins that are produced and fill it, instead of using what was described in a Radiolab podcast as a mugshot, you can put in a different let's say a faulty gene, the mugshot of a faulty gene, and it will work its way around and find copies of this faulty gene and cut it out.
Mm-hmm.
Because that's what it does, right? So and usually, it might cut this out and repair it, but if you have if you put in a replacement gene nearby, there are DNA repairing proteins nearby that will find where it goes and put it in its place. So
Wow.
And so this is how the mice become smarter than us, clearly, right?
So we may be able to inject people with stuff and fix problems that we couldn't possibly.
Well, I mean,
absolutely,
wow.
that's the hope.
Very
promising,
amazing.
but I mean, I'm sure that there is some physical limitations to this. I mean,
Well, it hasn't been tested in humans. The limitations
how do you
seem to
spread
be.
this all over?
Well, you can use probably I
variation
imagine a
across-
viral vector, wouldn't you?
Well,
But if that's taking it in one direction. For therapeutics, that's one thing. You might actually be able to correct sickle cell anemia or something, or some other inherited disease in the germline. Or let's say that you do it in vitro, you could do it in the eggs, and it would just be So I mean, you could
no more autism.
You could conceivably do that, but there's huge
that's
ethical
tricky,
issues that go on there, and I don't even think that people are going there yet. Although I know that they have modified some human embryos to show that it's possible. But the other thing is that this CRISPR technique works in every species so far.
Huh.
Every species.
This is
Because
basic.
DNA, it's the same program, right?
Wow.
It's the same code. And so you can,
that's both very cool and kind of scary, actually.
It's amazing. Either way, I mean, potentially, this is why I really want to understand this, because this has possibilities to be the big thing in therapeutics, the big things in GMO crops, and if you could bring up my first slide, in the control of these little fuckers.
Yeah.
All right. So what we have right now is an outbreak of Zika virus. This is a global pandemic. It looks like it's a public health emergency that's been declared by the WHO. We've been unable to allocate funds to attack it, and it turns out that there was a study that found that it may in fact attack adult neural stem cells, which
I saw that too.
So it looks like, which might explain why Guillain-Barré syndrome occasionally occurs in adults, the immune system attacking the nervous system, which is a very rare side effect of Zika, but seems to be associated with it, or symptom of Zika. But so this has been used. If you can add DNA at will to organisms, you can perhaps you could engineer an organism that selects itself out of the gene pool. What's the next image that I have up? I forget which one I put up next.
The giant mosquito eating people.
That means genetically modified mosquitoes.
Yay!
You could conceivably do this with
the big spiders better.
Keep growing. But so this group Oxitec, which is a UK outfit, have engineered a mosquito that needs to grow with the antibiotic tetracycline in its environment. It needs to have that in its system, or it will die.
Jurassic Park-ish.
Huh?
Except life finds a way. Life finds a way.
Exactly, I know. It's like, and then
Listen to Jeff Goldblum.
Yeah, tetracycline's going to be found in local plant, or it just feeds on people who are taking antibiotics. So this is that's one strategy that you could use to perhaps control a a population. You release male mosquitoes, they mate, and their offspring die because they don't have access to tetracycline. One way to do it. Another strategy is to release GM bugs that could only make male offspring.
I've heard that one before.
That one will, that will crash a population. I think you could probably do it in like 11 generations
Just as
or
long as
really
you're
quickly.
using frog DNA, right? Is that what
Yeah,
it is?
That's right.
Yep.
Well, and the females for the spiders too, right?
Yeah, and that's the thing. We don't have to worry about being contaminated with it, because we won't ever really come into contact necessarily with it. The other thing is that a third strategy that you could use this.
Forget that food chain stuff. I mean,
Well, yeah. The third way that you can use this is to engineer mosquitoes that are resistant to the diseases that make us sick.
Yeah.
And that would disrupt the life cycle of these little parasites. And we could conceivably get rid of malaria, Zika, bring up the next image, dengue fever, chikungunya. That's Zika.
It's pretty.
It is kind of pretty. There have been tests that have been done, and there's another thing. These reproductive solutions are, I think, what's known as gene drive technology, where you selectively favor even adaptations that are not beneficial. So they're designed to burn themselves out, which is kind of a wild concept. There have been tests in the Cayman Islands. 3 million genetically altered bugs were released there, and the aegypti mosquito, the one responsible for Zika and chikungunya, and I think dengue fever too, that population fell by 90%. It cleans itself up. There's no mess, and the first U.S. trials is set to start a couple hundred miles south of where the current outbreak is, in an island, Key Haven, I think it's what it's called. And there's, of course, you got to be. So, what are the potential environmental impacts of removing mosquitoes, right?
Mm-hmm.
I personally don't give a shit, because the sheer volume of suffering that has been caused by things like malaria. Fuck them, kill them all. Kill the invasive little bastards, or you know what? Don't kill them.
Just as long as there's nothing serious that causes it. Yeah.
Just interrupt the life cycle of that one disease, and then reintroduce the little fuckers back into the wild.
Yeah.
Or whatever. There are ways around this. These tropical diseases have killed and caused so much suffering, and we have a chance to stop that. I think that's an awesome thing.
Yeah.
We have seen 1.5 million infections in Brazil so far of the Zika virus, and 3,500 cases of microcephaly, which is the principal, which is the scary
so sad.
outcome. Yeah. It is a 50% of the human population is at risk of malaria. Let's end this. We could do it.
Been chronic for a long time.
Mm-hmm.
I just want to point out that, because I hear you like conspiracy theories.
Yes.
Genetically modified mosquitoes were released in parts of Brazil to counteract, I think it was dengue fever. And there are conspiracy theories that somehow those mosquitoes were responsible for Zika. They're not.
No, they can't be, because it's the males. The males don't bite.
I know that. But have you ever met conspiracy theorists?
Yeah, they do it. Yeah, have I ever met a conspiracy theorist? Yeah. There's things like facts that are, pliable.
Yeah.
But I mean, you don't have to have any basis in reality for them to find something. So.
So this is. I think that the CRISPR story is so cool. Really directly targeting genes. And it like the previous techniques would cost, to do one of these gene swaps, be several $1,000s. They say that the cost for this, which is already occurs in nature,
Which
is
is about
$75.
$30,000 after insurance and.
Yeah, but this is $75 to do. You could do
That's
home
couple thousand
biology, home genetics, people.
Oh my God.
Yeah. So.
Dial a treatment, and we all have these machines that just spit out what we need.
Replicators?
Yeah.
Yay!
Yeah.
I want replicators. So
I think
bad.
that I don't know.
Although it probably would cost more to run than my air conditioning.
Yeah, but maybe you could replicate air conditioners and.
But then I would have to pay for the air conditioner.
Fill your body with
that
antifreeze or whatever.
That didn't
Can
help
I just
the problem,
replicate
did
cold
it?
air? No.
That's a good. That makes sense.
Yeah, I'm just gonna throw some shade your way.
Yes, as you do.
Yeah.
Thanks,
Gotta
Bob, for that
replicate some trees.
fascinating story. And
Yeah,
putting up with
and
my
really the
inability
reason I
to
did
follow.
that was to acknowledge myself.
In knowledge.
Uh-huh.
In knowledge.
Because
I'm not sure that's
think
a word.
I should know what I should know what this stuff is and what it does and why it works.
Awesome.
Where it comes from. Yeah.
So that's the show. Thanks everybody for watching. Let's do our wrap-up stuff. Like, for instance, did anybody get the trivia question? Actually, I think somebody got it really quick.
Yeah, I saw Nathan get it really quick.
Yeah.
I saw Nathan get it really quick too.
Yeah,
Reiterate
so
the question and
the
the
question
answer.
was following today's this morning's earthquake in Italy. This is the anniversary of another geologic event, geologic catastrophe in the region. And what was that event? And it was the eruption of Vesuvius and the destruction of Pompeii and Herculaneum.
Yep.
It is there was a wonderful passage from Pliny the Younger, who was across the
Pliny.
bay, basically. His
Uncle.
uncle
Pliny the
went
Elder.
into town to try to rescue people, and he has this description where he explains. Where is that?
I don't know.
So, okay, they debated whether to stay indoors or take their chance in the open, for the buildings were now shaking with violent shocks and seemed to be swaying to and fro as they were torn from their foundations. Outside, on the other hand, there was the danger of falling pumice stones, even though these were light and porous. However, after comparing the risks, they chose the latter. In my uncle's case, one reason outweighed the other, but for the others it was the choice of fears. As a protection against falling objects, they put pillows on their heads, tied down with cloths. I just love that's that image of people running around with pillows on their heads.
Yes.
And that's all.
And
really.
dying anyway.
Yeah, well, bummer. But yeah, so
Yeah.
porn went out for Pliny the Elder.
Yes. So thanks, Nathan, for that response. Way to go. No bonus points. Sorry, dude.
Yeah.
And that's that. Announcements? Do we have any announcements? I don't see any announcements on my sheet. Anyone got anything going on this week that need to talk about and stuff? No? Flying sharks?
Nope.
I didn't ask this week if anybody was going to identify the video behind me. I just assumed that anybody who could possibly answer would try. Nobody tried. Which is, the thing about assuming. Anyway, Barbara got this in pre-show easily. She's so good at this. So if anybody can, in the next couple of minutes, maybe try to dial it in, I'll give you credit. But for now, book time, I guess. Everybody got some books? You got a book thing with a book? The little thing with the writing inside that you need to read stuff. Who's up? Barbara? Bob?
I'll go.
Go.
The use
Bob.
of the seal of the American Medical Association and the approval of products by that body was started by Boss Simmons back in 1905 during the early stages of his dictatorship. Strange to say, the racket was born not of a desire for money, but of a desire for revenge against a man whom Simmons thought had taken his girl away from him. It was Fishbein who later developed the scheme to the high degree of efficiency with which the seal is now mainly used. Man, there are lots of scare quotes in this book.
Wow. Is this
Was
some
that
kind of
part of the quote or part of the
yeah.
reading?
No.
Who's next? Eve?
Of
Gonna read a cat?
of all religions, Christianity is the one most concerned with dead bodies. Many religions have the idea of a holy man or woman, a living human being with extraordinary powers derived from special contact with the divine. Medieval Christianity developed to an extreme degree a distinctive form of this concept, the idea that the dead bodies of these holy people should be cherished as enduring sources of supernatural power.
Barbara?
It was Jo. I had never heard her scream like that in our life together, but I knew who it was just the same. "Stop hurting her," I shouted into the darkness. "Whoever you are, stop hurting her." She screamed again as if something with a knife, clamp, or hot poker took a malicious delight in disobeying me. It seemed to come from a distance this time, and her third scream, just as agonized as the first two, was farther away still. They were diminishing as the little boy's sobbing had diminished. A fourth scream floated out of the dark, then Sarah was silent. Breathless, the house breathed around me. Alive in the heat, aware in the faint sound of dawn thunder.
That sounds actually familiar. I had my mom's pork roast and homemade blueberry pie, baked with shortening and blueberries she picked herself. She didn't pick the shortening herself. I had a jet tila steak. I ate Italian food with the Prince of Sri Lanka. I ate Chinese food at the best restaurant in Beijing, not as good as the pizza in New York with Lou Reed. I've had scallops on a dive boat, brought up from the bottom of the sea and served distinguishingly fresh, almost live, with some wasabi right there on the boat. I've had my mom's macaroni and cheese with real Vermont cheddar. I've had one of Mrs. Teller's glorious meals with 3 different kinds of succulent meats and fresh bread and butter. I had the first Egg McMuffin served in Greenfield, Massachusetts when I was in high school because I thought it was funny to stay up all night and be there when they opened. What was your book, Bob?
My book was a weird-ass little thing by Morris A. Beale called Medical Mussolini, which is basically what the 1945 version of what Mike Adams is doing to David Gorski. This is the first really angry, incoherent rage blog.
Yeah, it's my first guess was some kind of alternate history thing. It was just really weird, but yeah.
It kind of is.
Yeah. What was your...
Why Can the Dead Do Such Great Things by Robert Bartlett.
Huh.
Why do the dead do such great things?
That's cool.
I've not heard anything great done by dead, but... Barbara.
Mine is Stephen King's Bag of Bones.
Hey, I thought that's what it was. I can usually tell Stephen King by at least some cues or some kind. Awesome.
It's one of the books. No, it's one of the books we read on our trip.
Ah, cool. I just got this one in the mail because I kickstarted another book called Our Broken Plate. This one was kind of one of the perks for the other book. This is Penn Jillette's Presto, and it's about his weight loss, which he lost like 100 pounds in like, I don't know, 3 months, 6 months, something like that. But I'm only just now getting into it. I suspect it's not going to be as good as the actual book, the science-based diet book that's supposed to be the one I supported. I know a few people that also know Penn that also lost weight by whatever the heck they did, which I haven't figured out yet, but I'm just curious and skeptical. But it's, Penn's funny, so it should be an interesting read.
He writes well, too.
Yeah. So there we go. That's the books, that's the stuff, that's the show. We did the thing and the thing at the place with the stuff and the people. Thanks for watching. Anything else?
No.
Oh,
Everything.
one last minute little thing. Nathan Miller is 2 for 2. He also got the AI on the TV. So that's what it is. It's artificial intelligence. Little creepy robot boy. So there you go. Logo, music.
Bye-bye.
Thanks for watching. Virtual Skeptics is an independent production of OnceTheHarm.net, Skeptical Humanities, myself, ICBS Everywhere, Doubtful News. Our logo was assigned by Sarah Mayhew at sarahmayhew.com. Our theme music is by Music Man and is user permission. I swear, the music is really low. Is that really low?
It's
Yeah, it's kind of
actually, we can actually hear you for a change.
There is that, yeah.
We usually can't hear you
Ah,
because
okay.
the music's too loud.
Good. Good. See ya.